Hi, and good afternoon, everyone. My name is Justin Malignoni. I'm a nurse practitioner, as well as one of the co-founders and chief strategy officers of RS BioTherapeutics. I'm excited to talk to you about our breakthrough therapy for COPD and lung inflammation, as well as share our investment opportunity. One of the reasons I created this company was because of my experiences bedside at dealing with COPD and other respiratory diseases, and seeing firsthand the burden current treatment regimens of steroids placed upon my patients due to multiple and significant adverse events. But first, let's talk about the COPD market. COPD market is massive. It is the third leading cause of death in the world, with incredibly high economic and social burdens. The US market alone has over 15.7 million people diagnosed with COPD and is projected to be an $8 billion market in 2023 alone, with a CAGR of 5.1%. There are a couple big players out of big pharma that dominate the COPD market. However, 4 of the top 5 products used in COPD each contain steroids. Steroids themselves, while reducing inflammation in the lungs, do so at a heavy cost of significant adverse events, including, but not limited to, increased prevalence of pneumonia and other lung infections, insulin resistance leading to type 2 diabetes, increased risk of bone fractures, and many more. These adverse events are so significant that this year, the International Guidelines for Treating COPD, something called the GOLD Guidelines, are no longer recommending steroids for initial therapy or follow-up therapy for the treatment of COPD exacerbation. This really hamstrings and complicates things for providers when treating COPD. But there hasn't been a breakthrough in treatment of lung inflammation in COPD in over a decade. Well, that is until now. It is my honor and pleasure to introduce RSBT-001, a first-in-class alternative to inhaled corticosteroids to treat lung inflammation, with COPD as our first target indication. So what is RSBT-001? RSBT-001 is a non-steroidal polarity-adaptive micelle that has been micronized to be delivered as a dry powdered inhalation product. Additionally, a composition of matter patent was filed in 2019. Now let's dive into some of the features and benefits that we have. First, inside this polarity adaptive micelle exists a proprietary cannabinoid complex that harnesses the immune modulating power of specific anti-inflammatory and non-intoxicating cannabinoids. This complex reduces pulmonary inflammation by targeting the inflammation cascade in the lungs at multiple levels. The metal coordination creates the polarity adaptive micelle, which creates the transport mechanism to penetrate the lung tissue, as well as deliver the active pharmaceutical ingredient directly into the cell. RSBT-001 reduces inflammation broadly across multiple biomarkers and offers us the ability to create a unique platform for future drug development as well. We plan on delivering this as a dry powdered inhalation, which is a highly effective and targeted way to deliver drugs to the lungs. We expect this to be well tolerated because of what we know about the components in this complex. Our animal and safety toxicology studies begin early next year to confirm this. And finally, there is no THC, so we have no concerns around the DEA or scheduling class of this product. Additionally, there are no concerns around intoxication, habit formation, or psychotropic effect. So how did we get here? Why do we know all this? Well, before we get into the data, I want to talk briefly about COPD and other lung inflammatory diseases. What you see here is a conceptual representation of the most common immune cell types and biomarkers relevant in inflammatory lung diseases, kind of like the biomarker equivalent of a fingerprint of the disease. It is demonstrated here by the colored squares in each of the disease. These are the markers that drug developers use to create new treatments for these diseases. This will become important in a few moments. Now on to our data. we conducted 2 separate animal trials at Marshall University with mice. What we are showing you here is just some of our data from our second larger mouse trial. The 4 markers you see represent macrophage and neutrophil activity at 24 hours. This data is after a single dose of either the insult, our positive control, or our product. The red bar reflects the inflammatory insult activity that we caused in these mice lungs. The purple bar represents one of the most powerful steroids in our arsenal, dexamethasone. It's given at ICU level dosings here. As you expect, you see the inflammation reduction pretty significantly. Now here is RSBT-001, represented by the green bar. As you can see, there is significant reductions across the board, even in comparison to the most powerful steroid in our arsenal. Here again is just some of the data from our second trial. We are happy to go through all of our data with any potential investor. However, what you see here are 4 of the significant inflammatory markers and their related diseases listed out. As you can see here, yet again in the red, is the insult, the inflammation that we caused. The purple, again, is dexamethasone. And finally, again in green, is us, RSBT-001. As you see, yet again, our compound was more effective in reducing inflammation than the most powerful steroid in our arsenal. Finally, given these results across 2 separate trials, we expect RSBT-001 to likely be a once-a-day medication. Now, when we look at the fingerprint, wherever you see a check mark, indicates that after review of all of our data across 32 biomarkers, we were non-inferior to the strongest steroid in our arsenal for reduction of those biomarkers in the lungs of mice. Again, keep in mind, dexamethasone is more than 5 times more powerful than prednisone, the steroid you may be familiar with. Now, we of course were incredibly excited and thrilled with our biomarker results around COPD, but we weren't the only ones. We had the opportunity to circulate our findings around the NIH. After they reviewed our data, the lead investigator in pulmonary fibrosis disorders came to us, asking if we would be open to a fully funded research collaboration to explore our compound and not only the treatment of idiopathic pulmonary fibrosis, a rare deadly disease affecting 140,000 people with no cure, but also explore RSBT-001 as a prevention for fibrotic lung changes. Outside of the significant amount of good this would do for those affected by this terrible condition, it also has very significant business implications for us, which can be summarized as big pharma likes rare disease assets. Some of the reasons being are listed out here. One of them is the fast-track review through the FDA. Another is 7 years of exclusivity from competition in the idiopathic pulmonary fibrosis market. This all leads to a significant change, both in terms of valuation and potential exit opportunities. So we have shown you our incredible preclinical results. However, that's only one variable in the equation for a successful company. The other variable that is just as important is the team to get it across the finish line. We have created an all-star team out of the pharmaceutical industry. Just some quick highlights of our team. Our CEO and leader is Dean Hart, who has spent over 40 years in the pharmaceutical industry, including 2 multi-billion-dollar brands under his belt. Our CMO is Dr. Michelle Shuppet, who is part of Spiriva, one of Boehringer Ingelheim's top multi-billion-dollar COPD products. Our CFO is Curtis Ryan, who has extensive history in pharmaceuticals as a senior vice president of finance of Takeda Pharmaceuticals. He was with the company from launch as it grew its annual revenues to $3.5 billion, as well as the CFO of Ovation Pharmaceuticals, where he helped sell to Lundbeck. We are also happy to have an amazing team of advisors who are helping us with business strategies and regulatory considerations. Now, on to the opportunity. We had completed our seed one raise last year for $2.7 million. And you can see the things that we have accomplished listed out. We are now in our seed two raise and are targeting $10 million. We already secured $1 million so far this round. These funds will be used to fund our animal safety toxicology trials, manufacturing costs, operations and our expenses for filing our IND with the FDA, and after its approval, begin human clinical trials. We hope you choose to join us on this amazing journey. We would be happy to set up additional conversations and answer any questions that you might have. Thank you very much for your time, and I'll be putting my email address here in chat.